Hello. Good afternoon, sir.
Good afternoon, sir.
Hello, Dr. Surya Prakash, sir. Good afternoon.
Good afternoon, sir. Good afternoon, sir. Can you hear me? Hello.
Sir.
Suman Kumar, may I audible?
Yes, sir. Yes. You are also audible, sir. Surya Prakash, sir. You are also audible, sir.
Okay, thank you.
Yes, we will wait only for 2 minutes.
Okay, sir.
Hello, Dr. Silke, sir. Please introduce the Dr. Surya Prakash, sir.
Okay.
To the participants, sir.
Good afternoon, all of you. First, I would like to welcome today's expert speaker, Dr. Surya Prakash Yadav. He is now doing his PhD at IARI, Delhi. He has qualified his MSc from IARI. Surya Prakash Yadav, he has ranked JRF rank 6, whereas he has ranked 5th in SRF. That means he has a huge experience and the study regarding all the preparations of JRF, SRF as well as JRF studies, because he has net qualified both UGC and ICAR, and I wish him best wishes for his future life. On behalf of the course director and on behalf of all the students and the faculty, I heartily welcome you, sir, for this online training program that is organized by Postgraduate Institute, Mahatma Phule Krishi Vidyapith Rauri. So, I request today's speaker, Dr. Surya Prakash Yadav, sir, to please proceed for your lecture.
Thank you, sir. Is this screen is visible?
Yes, screen is visible. You just make it okay. It's okay.
Okay. A very good evening to all of you, and I welcome you all. As Sir have introduced, I am Surya Prakash Yadav, and I am going to tell you about SRF levels regarding agrochemicals. So basically, natural resource management that contain four things: yes, agronomy, soil science, agriculture, chemical, water science, and technology. And here I am dealing basically with the agriculture chemicals. Agriculture chemical contains actually 11 units, and in different 11 units there are some important topics which I will discuss, and what are the important questions and what are the important things you need to cover. I will also discuss that. So I will start with unit wise. Actually, this agriculture chemical is very broad subject, and the 11 units needs to be covered in its take particular 2 to 3 months if you're going to study, and I need to cover in 1.5 hours. So I will tell you about the brief, what you need to study and what are the references you need to look after it. So the first unit is general chemistry. The first unit is general chemistry. Like in surface chemistry, why surface chemistry, and what are the things that you need to study? Like in surface chemistry, adsorption is important. Adsorption is a surface phenomena. Absorption is important, that is bulk phenomena. And what is sorption? That is adsorption plus absorption. And in this surface chemistry, you need to study about the Leach-Adarial principle. Basically, what it tells about how temperature and pressure affects on chemical reaction. Like if temperature increases, the adsorption decreases. And if the temperature decreases, the adsorption process also increases. Similarly, with the pressure, which is directly proportional to the adsorption phenomena. So you need to study about the Leach-Adarial principle and what is the effect of temperature and pressure over here. And also in surface chemistry, you need to study about the permanent gases and easily liquefiable gases. Like permanent gases, that is, that oxygens, nitrogen, hydrogen, these all are permanent gases, but they show less absorption on any surface. And easily liquefiable gases like ammonia, HCl, chlorine, they have higher molecular weight, so they tends to absorb more on a particular surface. You need to understand this, what is this all, and how this absorption is taking place. This is the important part. And basically, when we are covering for SRF for soil sciences student, all the topics written are not as much as important. Basically, the reaction mechanism part, they asked for a chemical student, but for a soil science student, it's, if you cover by surface horizontal, that is more than enough. And in adsorption, there are two types of adsorption, like physisorption and chemisorption. Like physisorption, it's a multi-layer phenomena, and chemisorption is a monolayer phenomena. Physisorption is reversible, and it's depend on absorbate. So you should know the difference between physisorption and chemisorption, and which is reversible phenomena and which is irreversible phenomena. These questions has been already asked, and the future chances they will also ask like that. What is the difference between physisorption and chemisorption? And pH is one thing that is you study in soil science also, and here we will also study in chemicals, like how different chemicals when added in soil, how they impact other properties of soil. So pH is important. And buffer solution, in buffer solution, acid buffer and basic buffer, you have just to remember what are the examples of it. You need not to remember all those things. What is the mechanism behind how they are buffering and controlling the pH of soil solution. So buffer solution, all the weak acid and strong acid, go for examples only. And these redox reaction, chemical kinetics, stereochemistry, and chirality, these are not much important as per soil science student. This is important for chemicals student. And other topic that is more important is nomenclature of organic molecule. Like you have already studied in class 12 that butane, ethane, propane, when some functional group, some simple types of functional groups are added in particular chain. So you should know about how they are nomenclaturing that. And SN1, SN reaction, two reaction, this is not important for us. But here, different reaction I have highlighted, like Claisen condensation, condensation reactions, Patonov-Bucci reaction, Barton reaction, Unpolong reaction, that is also called benzoin condensation reaction. So in this, what you need to study, what is the reactant and what is product. Like in Claisen condensation, ethyl acetate when react with sodium ethoxide, it forms acetoacetic ester. And when something is reacting with something, and is there any, you should look that, is there any byproduct is there or not. Like in this case, in Claisen condensation reaction, there is a byproduct of ethanol. So you should remember that only. No need to remember the structure, no need to remember the mechanism, but you should know what is the product and what is the main byproduct. And similarly, in backbend reaction, you will see that oxime when reacts with sulfuric acid, it forms lactone or amide. It has been asked what is formed. And in Curtius rearrangement, what happened, that azide when heated, it rearranged to amines. That's only you have to remember. And in Colbe-Summit reaction, you need to remember what is the main product. The product is salicylic acid, that is also used as a painkiller. That's why you have to remember the name and as well as the product. And in Michael reaction, you have to study this is in which there is a one donor and one acceptor, and the form product that is based on the addition reaction. And in Wittig reaction, Wittig reaction is like alkyl bromide when react with something. In this, the catalyst is more important, like triphenylphosphine. In the presence of triphenylphosphine, alkene is formed. So you need to remember that. In Wittig reaction, you need to remember TPP only. And no need to remember what is the reactant and product, but the main catalyst, you need to remember that. And in another reaction, Norris 1 and Norris 2 reaction. This is alpha bond cleavage reaction and alpha beta bond cleavage reaction. You need to remember only this thing, that in Norris 1 and Norris 2 reaction, there is a reaction in the presence of sunlight. So you need to remember here sunlight is used as a catalyst. And in Hoffman-Lefferty reaction, there is a haloamides. This is also this reactions also happen in the presence of sunlight, and amines are formed. This is all about. First, you need not to cover all this point that I have not highlighted, but the points that I've highlighted, you need to study that particular. Now coming to unit second, that is chromatography and spectroscopy technique. The first unit is not much important, and unit if I talk about that, on which part you have to more focus, this is the part where more questions are coming in exams, like what is basic principle and applications of chromatography, different chromatographic techniques, ion exchange, gas liquid chromatography, and HPLC, UV FTFI. So let me tell you what are the things that needs to be covered in this part. So first of all, on what basis it is, on what principle it is based. So generally they ask on the basis of partition chromatography, what are the instruments are based, like GC, HPLC, LC, these are based on partition chromatography. So this you just go through it. And second is, what is the instrument which are based on adsorption chromatography, like TLC and column chromatography. These are mainly important. Now coming to thin layer chromatography, they ask what is the stationary phase made up of, like silica oxide. You should know all the stationary phases and mobile phases of different instruments. Like in stationary phase, silica oxide is used, and generally there is a RF factor. So you need to know what is RF factor. RF factor is the value is there is a separation based on RF factor, and the formula of RF factor is distance traveled by organic molecular compound divided by distance traveled by solvent in mobile phase. So you have to remember the value of RF, the range of RF, and what is the main purpose of this. And any instrument, if you are using, what is its advantage and what is its disadvantage. Like in case of thin layer chromatography, the disadvantage is like the identification of similar compound is very much difficult. So some instrument had advantage and some other have not. So what is stationary phase made up of, and what is mobile phase? So if we talk about the thin layer, the stationary phase is silica oxide, and mobile phase like DCM, chloroform, methyl. You need to remember this. And in case of like paper chromatography is also like thin layer chromatography, and this is for color compounds. And there is something like ascending paper chromatography, descending, radial, 2D, ascending, descending. So you need to remember what is the movement. In ascending, there is a movement of upward movement of solvent, and in descending, there is a downward movement, radial like circular movement. You will get it in this after reading 2 or 3 times, and how this works. And in HPLC, in all the particular topics, HPLC is the most important. HPLC, first you need to remember what is the SPLC full form, like high pressure and high performance liquid chromatography. What is the pressure inside this HPLC? 50 to 350 bar. This is the most important point. Multiple times it has been asked. And the column of SPLC is called Herd. And the stationary phase, in stationary phase particularly, you need to remember the particle size. What is the particle size in HPLC? So basically the particle size varies from 2 to 50 micrometer, or less than 50 micrometer. You need to remember this. And what is the mobile phase of HPLC? Like it is polar compound, and stationary phase it is non-polar compound. In all the instruments, you need to also remember this. What is the detector used? Like in case of HPLC, UV visible detector used, PDA detector is used, MS is used. So you need to remember all these things. And there is a gradient and a constant composition of that particular mobile phase. So sometimes it has been asked that what is isocratic mode in HPLC? So this is when the mobile phase composition is constant, it is called isocratic mode. And gradient when the mobile phase composition vary. So this is called gradient mode. Now, There is a gas chromatography, gas liquid chromatography. This is based on partition chromatography. Here the carrier gas is mobile phase, like carrier gas is mobile phase, like here nitrogen is used, helium is used, but carbon dioxide is used, but oxygen is not used. So sometimes they give which of the following gas is not used in GLC. So you need to remember that oxygen is never used. And in stationary phase GLC, there is a polysiloxylone. Polysiloxylone is a compound which is used in GLC as a stationary phase. So this you need to also remember. And what is the function of gas chromatography? Its function, basically, it's detect non-polar and volatile compounds because the temperature is very high. This works in very high temperature. So you need to also remember what is the temperature of particular instrument, working temperature, like 350 to 500 degrees Celsius. This is very high temperature. And when any solvent is get into it, it's volatile and get detected. And the detectors used in particular gas chromatography is TCD detectors, ECD detectors, FID detectors. You need to learn all these detectors in particular. This is a little bit complicated because different detectors is used in different instrument, but the question is already asked from these types of detectors. Now, mass spectroscopy. Why mass spectroscopy is important? Because the detection limit is very much important, and they ask what is the basis of this mass spectroscopy. So generally the identification is based on mass by charge ratio. You need to remember this. And the high resolution mass spectroscopy is there, and in this the ionization is made by. So they ask what by which mechanism or by which device the ionization is made. It's chemical ionization, it's electron spray ionization, it's MALDI, or it's EI. So you need to remember this. And they also ask what is Mac-Lafferty rearrangement associated with. This is associated with mass spectroscopy. You need to remember this also. And there is a gamma hydrogen removal. Gamma hydrogen removal is there in that. And in spectroscopy, UV visible is important, IR is important, and NMR is important. In UV visible spectroscopy, there is an interaction of light with matter. And accordingly, based on Lambert's-Puyat law, it's follow. And you need to remember what is the range of UV. Like UV, there is a less than 350 meter, and where deuterium lamp is used. And in visible region, tungsten is used, in which more than 350 nanometer light is used. And this is more than enough for this particular topic. And coming to the next, chemistry of natural product. This is also very much important. Chemistry of natural product, particularly classifications, structure, chemistry, carbohydrates, protein, amino acids, enzyme. Many times we are familiar with this because we have studied in class 11, 12. So the basic question is already asked from carbohydrates, amino acids, enzyme, nucleic acid, what is vitamins, and their chemical names, generally retinols, like the lipid polymer. These are easy getting questions. You will get it. But juvenile hormone, molting hormones, these you need to study from this particular. Let me elaborate something on this. Actually, I missed one point in previous one. In this spectroscopy, in the second unit, you need to remember one more thing. There is an internal standard used in NMR, that is tetramethylsilane. The delta signal is 1. You just go through benzene, toluene, and phenol, and tetramethylsilane. What is their internal standard? And they are used in NMR. You also have to study this point in this. And what is delta shift? Sometimes they ask what is delta shift? Delta shift of particular chemical, delta shift of like acetone. What is his delta shift like acetone? It's 2.5. So you need to remember the delta shifts also. In this unit, carbohydrate, you have already studied what is carbohydrate, how they are classified: monosaccharides, polysaccharides, oligosaccharides. So they generally ask what is oligosaccharides, what is the number of units in there, and what is anomeric carbon, alpha anomer, beta anomer, what is epimeric carbon, that epimeric carbon like C2, C3, C4 carbon. The C1 carbon, when the difference is related to C1 carbon, it's called anomeric carbon. When it's related to someone like other than C1, it's called epimeric carbon, like glucose mannose, it's C2 epimer, glucose galactose, C4 epimer. So for this one reference book is there, like NCERT you have already studied, and in class 11th, the biomolecule chapter. From that particular chapter, you will get all the points related to it. And polymers, there is a polymer also chapter. I have given the reference book and later. And here you need to remember juvenile hormone, malting hormone, plant-derived nutraceutical. Now this is not much important. But the juvenile hormone, what is the example? Basically, you have to remember the example. And what is terpenes? Basically, they ask what is terpenes in chemicals. So when basically all the examples of terpene, they contain the carbon in multiple of 5. Like the monomer, if it is isoprene, it will contain at least 5 carbon, the isoprene, and multiple of that. If terpenes is added to oxygen, it's called terpenoid. So you need to remember that. And you need to remember the examples also, like monoterpene, it's 10 carbon. Sesquiterpene, they ask what is the number of carbon in sesquiterpene, like it's contain 15 carbon. And diterpenes, it contain 20 carbon, triterpenes 30 carbon, tetraterpenes 40 carbon. And the examples had already asked what is this lycopenes. Lycopenes is tetraterpenes, so you need to remember this. And in alkaloids, they ask the definition of alkaloids. They are basically secondary metabolites, they are nitrogenous compounds, they are basic in nature, and they are biologically active. This I have already asked. And they contain more than one nitrogen, and they contain heterocyclic rings also. You need to remember, you need to study terpenes and alkaloids in little bit details. And example of it, what are the plant derived alkaloids, and from which part they are derived, and how they are used. What are the products formed from these compounds. In juvenile hormones, like these are acyclic sesquiterpenoids, so they prevent metamorphosis of insects. So you need to remember the example, like methoprene. Methoprene is example of juvenile hormone. It has been asked already. And what is anti-juvenile hormone? Reversible of that inhibit the juvenile hormone synthesis, like protein 1 and protein 2. You need to remember also this. And malting hormone like ecdysone, alpha and beta. You need to remember all these things. Coming to Unit 4, this is also very much important, like naturally occurring insecticides, natural pyrethroids, nicotine, retinol, neem, currant base. This is very much important. And semiochemicals, when we have given our exams, the definition of semiochemical has been asked, and it is much confused by alkaloids definition. So, and phytoalexin definition. You have to remember all these definitions of phytoalexins, semiochemicals, advantage and limitation of natural pesticides, and chemosteroid. Example with definition. In coming to pyrethroids, they are synthetic, and pyrethrin, that is natural. So you have to remember the cypermethrin example, elethrin example. Elethrin is also used in all-out mosquito killers. So you need to remember this. Fenvalerate, this is amino acid containing pyrethroid, so you need to remember this also. Pyrethrin is a natural occurring insecticide. It contains 144 isomers, so you need to remember when there is something special associated with particular product, you need to remember. And how this is extracted, the extraction of pyrethrin is done by kerosene oil, and this is extracted from a flower of Chrysanthemum cinerifolium. So you need to remember. And what is the mode of action? The mode of action is neurotoxic. So you have to also remember what are the photostable synthetic insecticide derived from these particular chemicals, like permethrin. This is first photostable. You need to remember also this. And in case of retinone is called the fish poison, and it derives from the roots of Deres elliptica, Longocarpus. So you need to remember the plant's name and what is the functional group. Functional group is very important in some of the compounds, like chromain. Chromain is already asked in many of the exams, like chromain is a very functional group present in retinone. And what is LD50 value? LD50 value of, you have to remember only of those chemicals which have LD50 value very lower. And what is the mode of action? Like in the case of retinone, the mode of action is they inhibit respiration. They basically disturb electron transport system. And coming to nicotine is a extracted by aqueous alkali. Aqueous alkali, then steam distillation takes place. So you need to remember how they are extracted, what is the mode of action, and what is their example. Is there any photostable compound? What is the LD50 value? All of this. Like in case of nicotine, the LD50 value is 50 milligram per kg. And LD50 value means that they control 50% of the populations. And nicotine, when it's oxidized, it forms nicotinic acid. And nicotine sulfate, this is synthetic and it's widely used. And they basically, nicotine sulfate is mode of action is acetylcholinesterase blocker. They basically, acetylcholinesterase is an enzyme which generally degrade acetylcholine. Acetylcholine is a neurotransmitter. So they disturb acetylcholine. So mainly they target the nervous system. Now, neem-derived product like azadirachtin. Azadirachtin many times asked, what is azadirachtin? In terms of chemical, they ask, this is tetra-non-triterpenoids. So you need to remember it. Tetra-non-triterpenoids, azadirachtin is, this is non-toxic and its LD50 is much, much higher. It's antioxidant and growth retardant. You need to remember. And azadirachtin also inhibit nitrification. This is important for soil science, but for chemical point of view, you need to remember tetra-non-triterpenoids in case of azadirachtin. And what are the parts from which it has been derived? Like in case of seed, azadirachtin A, flowers, it has been derived like flavonoids, heartwood, nimbolene. So neem, different parts of neem, different chemicals is derived, and you need to remember all of their names. And azadirachtin is also called organosulfur compound. So you go through it once. And coming to Karanj, Pongamia plants from which, and the functional group, like I already tell, functional group in some of the cases is important. Like here, the functional group is chromine. Chromine is important here. And in case of nicotine, in case of retinone, the functional group is chromine. There is an importance. Chromine, I mean, single bond is there. Chromine, there is a condition of unsaturation, double bond may be present. So you need to remember, Karanj contain chromine and retinone contain chromine. This is also derived from seeds. Now coming to allylochemicals. Allylochemicals, they are secondary metabolites, and there are different types of allylochemicals. Once in our exam also, it has been asked, the term allylochemicals is given by. So allylochemicals terms is also given by Hans Mollis. So you're not confused, and also studies this. And what are the types of allylochemicals, like allomones, caromones, cinnamones? Like it's depend upon benefit to sender or benefits to receiver or benefit to both. So you need to remember the classifications of allylochemicals also. And plants from which allylochemicals are derived, like from gallic acid that inhibit growth is from spurge plant, euphorbia. You need to remember this. Like for coffee, mustard, sorghum, all the allylochemicals that are extracted. And here one example is important, like sugar beet. From sugar beet, we extract acetaldehyde. So acetaldehyde is important, mustard sinigrin is important, gallic acid. Here spurs, this is important. And in case of semiochemicals, I mean, chemicals, if you classify further, it's contain pheromones and allylochemicals. Who has given this term is also important. Pheromone term given by Carlson and Lucian. This is important. Allylochemicals, these are ectohormones. So these are also important. And what are their types? Pheromones are chemical released from insect to induce a response to another individual of same species. There is a term, same species. This is a key point. If this changes, so you just read the options very carefully. And pheromones are unsaturated and they contain even number of carbon. For a chemical point of view, this is very much important. Like in case of gypsy moth, gyptol, dyspalure, this is example of it. So you need to remember what is the example here. And chitin synthesis inhibitor. Buprofezin is an example. This is very much important example. Chitin synthetic inhibitor. Buprofezin, you need to remember here. What else point? For chemosterlant, in our exam, it has been also asked, what is TIPA? TIPA is an alkylating agent. It is a chemosterlant. You need to remember this. TIPA, thiotipa, afolate, metaTIPA. So you need to remember, and anti-metabolites, you need to also remember this. So this is a much important part, and it can 3 to 4 questions already asked in different exams from this particular unit. So you go in detail for this point. And phytoalexin, I think I missed phytoalexin here. The concept of phytoalexin given by Muller and Berger. This is important, and these are wording of compounds. They have basically broad spectrum application, and the molecular weight is low, and they are synthesized and accumulated in plant after exposure to abiotic stress or any microbes. So you need to remember when they are secreted. They are secreted when the plant is exposed to certain external agents. Only then they are secreted. So all phytochemicals that are plant, these are basically a defense of plants. So which plant remove which phytoalexin is very much important. Like in grapes, garlics, grapes, river stock is very much important. In garlic, alexin is important, and in mustard, camelexin. So you need to remember all the examples of particular plants and their released chemical in that. And macrolides. Here, I think macrolides is there. Macrolides, they are like ivermectin, milbemycin, from which these are extracted, and what is their mode of action is very much important. They are GABA agonist, although, but you need to remember from which they are extracted. Like ivermectin is extracted from streptomycin ivermectin. You need to remember the name. And Unit 5, this is also much important, like synthetic insecticide, fungicides, nematicides, and rodenticides. Insecticide, you need to remember what are the different types of insecticide. Contact, synthetic, you need to remember the example. Mainly the three groups are there: organochlorines, or carbamates, organophosphorus. So you need to remember what is organochlorines. Organochlorines, particularly DDT, you need to study in detail. Who have synthesized it, and a book is written on DDT ill effect, like Silent Spring. This has been asked in many exams, and many times it has been asked. Rachel Carlson has written it. So you need to remember the writer and book Silent Spring also, and what is its LD50 and why it is banned. That you need to remember the reason behind it also, and which form of DDT is toxic to plants, and how it is changes in different form after changing its particular functional group position. In like HCH, which is also called gamexine, or this is discovered by Linden. So its biologically active form is gamma form. So you need to remember whether it is alpha form, whether it is beta form, or whether it is gamma form. So in case of HCH, gamma isomer is very much important because it is biologically active, and you need to remember the most stable form also. Like in case of HCH, the beta form is most stable. So you need to remember all these things. In case of endosulfan, you need to remember in our exam stars, it has been asked when did endosulfan ban and what was the reason behind it. Basically, it has been asked on the name of Kerala tragedy. In Kerala tragedy, there was endosulfan tragedy. So it has been asked. And what is the mode of action? Organochlorines are basically neurotoxic. So you need to remember this also. And Linden work as a GABA receptor attack. They also attack on nervous system. Now, some question has been already asked about the persistence of any organochlorines and organophosphates and carbamates. So you need to remember that organophosphate or organochlorines are much persistence in soil than carbamates, than organophosphorus, because due to halogen group, the persistence of particular organochlorines is much there. And you need to remember the examples of all or carbamates. Like carbamates, one example is LD carb. This is much important because this is most toxic. Its LD50 value is 0.93 milligram per kg. So LD carb is most important. And what is the mode of action? Like it's block acetylcholinesterase. Achhe blocker. It's also written like that. ACHE blocker. So you need to remember this also. So acetylcholinesterase blocker. And one more example, like Begon, that is Propaxer. that is contact type. And in case of organophosphorus, there is one difference between carbamates and organophosphate. The activity of carbamates is reversible, and the activity of organophosphorus is irreversible. Like there is no sulfur compounds, here in carbamates, but in organophosphorus there is sulfur compound mix. So what is the difference? They also ask the persistence. They also ask and number of sulfur compounds in different organophosphorus. You need to remember what is the derivative of thiophosphoric acid. Like example, parathion, quinol phos, chlorpyrifos, stock. So basically they ask which of the following is derivative of thiophosphoric acid. So you need to remember 3 to 4 example, and how many sulfur it contains. Like parathion, quinol phos, it contains only 1 sulfur. So you need to remember. There is some exceptions also, like in case of dithiophosporic acid, they basically contain 2 sulfur. But the example of dithiophosporic acid is 4-8. That contain 3 sulfur. Number of sulfurs is 3. So you need to remember also this. And there is estramide of phosphoric acid, is acephate. In chemical for soil science student, the examples are much important of particular chemicals. Examples, definitions, mode of action, and their persistence in soil. So you need to remember all these things. When we talk about fungicides, like inorganic fungicides and organic fungicides, you need to remember about Bordeaux mixture, Burgundy mixture, copper oxychloride, their mode of action, and what is organotin compound, what are their examples, what is organomercury compound, what are their example, and what is the mode of action. Like organomercury compound, they basically act on respiratory system, and they block respiratory enzyme to function. So metabolic activity generally stops. And you need to remember also what is organic fungicides, like organic phosphorus, ketazine is there, adifenphos is there. Only 1-2 example you remember. And dithiocarbamates group like nebam, zinc containing, magnesium containing, any particular element that is associated. Like sometimes they ask what is zinc containing, zinc, magnesium containing, manab. So you need to remember all these things. And azole compound in fungicides also there is a azole compound you need to remember, and the example is benomyl. You need to remember the benomyl name. And PBO is asked in 2018. What is PBO? PBO is a synergist. The options were synergist, chemostimulant, and phytoalexin, like that. So PBO is a synergist, and this is fipronil butoxide. So you need to remember this also, the example of synergist. And safrole is another example of synergist. And in case of rodenticides is also important. Why it is important? Because their mode of action is anticoagulant. Like in case of warfarin, many times it has been asked warfarin. This is widely used, and the warfarin is basically an anticoagulant. And other examples is N2A-NTU. This is ANTU. This is also rodenticide. So you need to remember this. And crimidine. Most important is zinc phosphide and warfarin, because zinc phosphide is inorganic and warfarin is plant derived. It's animal derived. So, and it is widely used. And another thing is about nematicides. Nematicides, you need to example what is its example, like methyl bromide. The usage is banned. Why it is banned? They generally ask question why methyl bromide usage is banned. Earlier it has been widely used, methyl bromide, CS3Br, because this halogen group, what it do? It's degrade our ozone layer depletion. Ozone layer depletion is there. So because of that, this chemicals had been banned, and now other DD chemicals had been used. Nemagen is another example. Now, 2 chemicals you need to remember. What is botanical derived nematicides, like bisphernil and terphenyl. These are derived from marigold plants. So you need to remember what is the example and what are the plant derived nematicides in this particular section. And now coming to unit 6. Unit 6 is herbicides and plant growth regulators. In herbicides, you need to remember particular groups like phenoxy acid. What is the example of phenoxy acids? Like 2,4-D, MCPA, 2,4-T, Agent Orange. Basically, they ask what is the composition of Agent Orange, like 2,4-D and 2,4,5-D, because it has been also used in USA and Vietnam, and it is very much popular. So they ask this. And growth to death, growth to death is related to whom? So it is related to 2,4-D. So you need to example particular classification of herbicides, their examples, and in halogenated, there is TCA is important. TCA and Delapon is important. Delapon white is important because it is used for aquatic water weed, and amide group. All the amide group are very much important. In all these things, amide group is very important, like butachlor, alachlor, propanil. These are pre-emergence and systemic, except propanil. Propanil is a contact and post-emergence. So you need to remember which is exceptional part of any particular group and what are their example and what is their mode of action. Like amide group, the mode of action is protein synthesis inhibitor. Coming to triazine group, triazine group examples is very much important because when cyanuric chloride react with alkyl amine, triazines are formed, and you need to remember which is symmetrical triazine and which is unsymmetrical, asymmetrical triazine. Simazine and atrazine, these are both are symmetrical triazines, and metribuzin, this is asymmetrical, and propazine and chromazine, these are first triazine compounds. So you need to remember, and you need to remember also this in triazine, which particular example has a highest water solubility or maximum water solubility. So it's atrazine. You need to remember this. That's 33 milligram per liter of water, and their mode of action is they inhibit Hill reaction, photosystem 2 reaction. So and one question is also asked related to triazine. What is the metabolism of S-triazine compound? This triazine compounds, GST, glutathione S-transferase. This is the metabolism by which they are breakdown. So this is much important from this point of view. And one is urea compound. Urea compound is also important. And urea, the basic examples are uron, diuron, monouron, isoproteron, phenuron, and their mode of action is they also inhibit photosystem 2. And Hill, they basically stop the Hill reaction. And diuron, you need to also remember the plants in which it is used. Like in case of diuron, it is used in cotton. In case of monouron, it is used in oats. And in case of isoproteron, it is used in wheats. So you need to remember all these things. In case of dinitroaniline group. Dinitroaniline group is also very much important. Like the examples many times asked, like pendimethyline, flucloraline, triflucloraline. These are pre-plant incorporation, like flucloraline and triflucloraline. And their mode of action is they generally inhibit the growth inhibitor. They check the mitosis activity, the DNA doubling, and all those things you need to remember here. And coming to bipyrrolidium group. Bipyrrolidium group, this is also much important because these are also called quaternary ammonium salt group. In the examples is very much important, like paraquat and diquat. And you need to remember what is the trade name. What is the trade name of that particular? Not all of that, very popular, which are very popular in market. You need to remember of that. And these are contact, and the mode of action of quaternary ammonium salt and bipyrrolidium group is little bit different from this particular triazine group and urea compound, because they inhibit photosystem 1. They are oxidant and block electron transferase activity. So sometimes in match the column and in objective question they ask what is the match the following, and which of the following is photosystem 1 blocker, which of the following is photosystem 2 blocker. And so you need to remember this, and the mode of actions too. Now coming to organophosphorus compound. In organophosphorus compound, glyphosate is very much important. You need to study what is it's derived from. It's derived from glycine amino acids. They basically they block tryptophan, tyrosine, and phenylalanine. These are aromatic amino acids. So you also remember which are basic amino acids, which are aromatic amino acids, which are neutral, and what type of amino acids is blocked by glyphosate. They are in systemic in nature, and what is the mode of action? Mode of action, as I've already told, like ESP is synthetase inhibitor. They generally inhibit aromatic amino acid synthetic activity. Means in the presence, when the glyphosate is applied, these particular amino acid will not form. So obviously the plant will not perform. So you need to study this. And one more thing is important related to glyphosate. What is the metabolism of glyphosate? In this metabolism, AMPA is very important. When glyphosate is metabolized by glyphosate dehydrogenase, AMPA is formed. AMPA. Some metabolism products are also important. This is amino methyl phosphonic acid. Amino methyl phosphonic acid, you need to remember this. And one is sulfonyl urease compound. Sulfonyl urease compound have low mammal toxicity. These are most widely used among all herbicide group. You need to also remember this. This is most widely used group among all these things, because the examples, when you listen, you get it. It's like sulfosulfuron, chlorosulfuron, metsulfosulfuron, chlorimuron, and pyrosulfuron methyl, in which you need to remember methyl sulfosulfuron, which is used as 4 to 5 gram active ingredient per hectare. And in this particular group, mostly we use sulfosulfuron. So you need to remember the activity. And what is the mode of action of this sulfosulfuron urease group? The mode of action is ALS blocker. They generally inhibit formation of valine, isoleucine, and lysine, acetolactate synthesis activity. So these are ALS blocker. And earlier I have told that organophosphorus groups inhibit aromatic amino acid activity. So you need to remember what kind of amino acid is inhibited by application of particular herbicide, by sulfonyl urease compounds or by organophosphorus compound. Now, some of the herbicide safener, sometimes it is not much important, but sometimes question has been asked from this parts also. What is the herbicide safener? You just have to remember the following examples, like Na. Na is a first safener, naphthalene anhydride, or PHA, phthalic anhydride. This is also a safener. So you need to remember the examples. And closatomazole, this is also a safener you need to remember here. What is pro-safener? Pro-safener is basically initially a compound is there. When it is applied, so its biotransformation takes place. When its biotransformation takes place, it's turned into active compound. So you need to remember one example of pro-safener, like CPMI. CPMI, when it's an inactive form, an aryl maleimide, it's a intermediate product. When it's applied, its form maleimide. Maleimide is the active product. So you need to remember this. Coming to next units. This unit is a little bit longer, and you need to remember so much names, particularly example in unit 6. In agrochemical formulation, this is not much important from our points of view. And in this unit, generally, they ask what is the size and what is the type of sprayer, what is the basic formulations, and what is the basic concept behind pesticide formulation. You need not to study in details all about all the types of sprayer, what is that, and you need to just remember what is the carrier, what is the active ingredient, and what is the rate of application in particular, and what is the example of particular solid, liquid, and these formulations. So in case of type of formulation, they generally ask what is dusting powder, what is water dispersible powder, what is emulsifier concentration, what is water dispersible granules, and what is their active ingredient, what is the percentage of particular active ingredient. These are the points you need to remember. You need not remember all these tools required to measure nanoparticle amount, these things. But the active ingredient percentage is very much important, In dusting powder, or either like in dusting powder, the active ingredient is 1 to 10%. You need to remember also the wetting agent used. Like dispersol, serzol, ident 20. Sometimes it is asked that which of the following is wetting agent and which of the following is dispersing agent. So you need to know the difference between wetting agent and dispersing agent. But so you need to remember the examples, like CMC. CMC is already asked, like sodium carboxyl methyl cellulose. This is a dispersing agent. And dispersol is already asked. This is 2 to 3%, and it acts as a wetting agent. And what is the particle size? In case of like water dispersible powder, AI is 50%. In its particle size, it is less than 5 micrometer. So basically, water dispensable powder is a combination of pesticide dust is there, and dispersible agent is there, and one wetting agent is there. So you need to remember what is all this thing. And in sometimes one very important example is there is also a pH of particular that thing, the inert material is added to carry that particular thing. So you need to remember the pH also. And in case of EC, the AI is 50 to 85%. You need to remember this also. In EC, there is a pesticides, there is an organic solvent. What kind of solvent is used? You need to remember. And what is the emulsifier? Emulsifier generally cause what? They disperse the particular formulation when it is sprayed. And what is the role of organic solvent is help in dissolution of particular chemical. But they sometimes also are, what is the disadvantage of EC? Disadvantage, it has been asked one times, that disadvantage these are sometimes phytotoxic, so you need to remember. And what is granular formulation? In granular formulation, there are two types: extruded granules and non-extruded. Extruded granules are those granules when they disintegrate, when they come in contact with the water. As soon as they come in contact with water, they disintegrate. And in case of non-extruded granules, they takes time to disintegrate. So this you need to study and you need to remember. And what are encapsulated granules? What is their advantage? Because if we study the traditional things, the release is faster and the disintegration is faster in soil. So encapsulated granules were used in that. Release is slow, and basic their principles behind that to control release and release of the toxicant. And what are the advantages? Like they have low mammalian toxicity, low volatile, decrease in phytotoxicity, the leaching losses are less, and they regulate the release of active ingredient. This is the main points here. And what are the disadvantages? You need to also remember, like it has been seen that honeybees, they misinterpret that, and they just pick as a pollen. They misinterpret, like this is not like that, say, and they understood that this is pollen, and this is the problem with that. So you need to remember advantage and disadvantage along with that. And one is ULV. ULV is ultra low volume. Ultra low volume means if the volume of particular something is very low, then its toxicity also must be very high. I mean, the concentration must be very high. So you need to remember. And the example of ultra low volume, you need to remember, like Ricin oil. Ricin oil, it is a mixture of aromatic naphthalene and deuterex and aromatic hydrocarbons, vegetable oils. You not need to remember the compositions, but you need to remember the particular name. What is ultra low volume particular solvent? And another thing is used adjuvant in agrochemical formulation. Adjuvant. What are the different types of adjuvants? So you need to remember there are 8 types of adjuvants. You go through what I will tell you the reference book from where you need to study. There is 8 types of adjuvants, like surfactant, foaming agent, activator, hygroscopic, extender. You need to study. And the classification, like here, solid, liquid, and formulation. Like they ask dust granules, water dispensable granules, and water encapsulated granules. These are example of solid formulation. EC and ULV is example of liquid, and gaseous like aerosol, fog, smoke, and mist. These are example of gaseous formulation. In case of gaseous formulations, you need to also remember the size of fog, aerosol, mist. Sometimes has been already asked in different exams. Coming to next, pesticide residue and their dynamics in environment. In this, most of the things is not much important in our point of view, but pesticide residue, ELISA is important, ELISA techniques, pictures is important, LOD and LOQ is important. It has been many times asked in our exam. So, for pictures, you need to remember what is its full form. Pictures like solid. This is a solid phase. This is solid phase extraction method for detection of pesticide residue in food. If I have to tell this, which is the most important thing in this particular unit, it's ELISA and pictures. You need to remember what is pictures. Sometimes you will confuse by this term easy. they interchange. They interchange with easy, effective, and something else. Easy and effective, and this easy and effective here and there. So don't be confused. R is rugged. So this is quick, easy, cheap, effective, rugged, and safe. The application is broad range, and it is environmental safe. And why it is environmental safe? Because it has been also asked, because there is no halogenated solvent used. This is why it is safe. So they ask generally what is questions, what is the mode of their extraction, and what are the procedures. Procedure is something deep question, something, but you need to remember there is a DSPE, that is dispersive solid phase extraction is there, and primary and secondary aryl amines were used in particular questions. And another thing is ELISA. ELISA is a pesticide residue estimation, and this is for unstable pesticides. So you need to remember what is the chromosome. What is the chromosome used in ELISA? This is very much important. Chromosome, particular example, para-nitrophenol phosphate. This is used. So this is very much important. And in pesticide residue, one extraction and cleanup technique is important. Here, extraction is, I mean, it's from plant, water, soil, or whatever. But the cleanup is co-extractive removal of pigments, oil, and fats. What cause interference? So cleanup is also very much important. For example, points of view. And in MRL, you need to remember what is LOD and LOQ. LOD is limit of detection, and LOQ is limit of quantification. You need to remember its full form. And what is it? What is signify? LOD is minimum level that can be detected, and LOQ is minimum the level which is detected and quantified at minimum level. You need to remember all these things in here. And accelerated solvent extraction technique, ASE. This is also important. Accelerated solvent extraction. There is a high pressure and high temperature extraction. So recovery is automatically more because temperature and pressure both are high in accelerated solvent extraction. As the name already suggests, it's so you need to remember there is a high pressure and high temperature. Sometimes the question has been framed that low temperature, high pressure, high temperature, low pressure. So you need to remember what is the temperature and pressure here, and not much important things are there in this particular unit. Next is agrochemicals regulation and quality control. Here you need to remember what are the different banned and restricted pesticides in this particular unit. You need to remember the recent data published, and you need to remember total number of registered pesticides, number of banned pesticides, formulation, number of pesticides withdrawal, pesticides, restricted pesticide. I mean, you can use it either in fruits or either in vegetables. You need to remember here, and number of biopesticides registered in India in this particular unit. These are most important, and you have to see it before exam because the data come, and you need not to study from here. It will, comes in March or maybe in April. So you download it and you just go through it. What are the types of pesticide that has been banned? What are the pesticide that has been, the formulation is banned, number of biopesticides, how much consumption of pesticides per capita is there, and where there is the most consumption of pesticide, in which crop there is a most of the consumption of particular pesticides. Now CIB and RC, this gives registration. There is 3 types of registration that you need to know, and the insecticide acts, this you also need to know. What is insecticide act, what is insecticide rules when it comes, and what is the date. You need to remember all these things. And if particular laboratory is set up, what is the center of that particular university, you need to also remember that. And in CIBRC, there is 3 types of registration is there: provisional registration, regular registration, repeat or me too registration. Provisional registration is for 2 years, regular registration is for permanent, and repeat or me too registration when sometimes registration of a particular company is failed, so they again apply for that particular things. So you need to know provisional is related to which, regular is related to which. So CIB and RC, This comes under Ministry of Agriculture and Farmers Welfare. So you need to understand this. And you need to remember what is the number of registration committee, how many, who is the chairman of that particular committee, how many members are there, and what kind of certificates they are providing. You need to study. And another thing is Environmental Protection Act. This comes under Ministry of Environment, Forest and Climate Change, and this has been formed after Bhopal gas tragedy in 1984. After that, it has been formed. So what is its functions, how it's related? So you need to study here. And one more thing, hazardous waste and its management and handling rule. It has been, it's in 19, it comes in 1989. So you need to remember what is the year. And pesticide management bill, you need to study also this. This has been amended in 2017 and enacted in 2020, and it replaced pesticide management bill, actually replaced Insecticide Act. So you go through it and study what are the changes they have made already in this. And in Environment Protection Agency, when it has been formed and why it has been banned DDT. This is the agency which has banned DDT in 1972, and it has 27 labs and 10 regional stations. So you also see the latest details about when we are giving this year exam for soil science. So you go for it. You just remember the latest data of all the labs, and in this particular unit, you need to remember all the latest data, because the latest data is always asked. And for say in India, Food Safety Standards Authority of India, this comes under Ministry of Health and Family Welfare, and its headquarters in New Delhi. It has been formed in 2011. You need to remember what is its chairman, who is chairman, how many members are there under any particular committee, and what is Food Safety Act. And there is total 8 acts in FSI. You need to remember what are the acts, like Prevention of Food Adulteration Act, Food Production Act, Meat Production Act, Vegetable Oil, Edible Oil. When it enacted particular year, you need to remember all these things. And the license provided by FSI is also important for chemicals point of view, like basic license. When if you are setting up a laboratory and your particular amount is not exceeding 12 lakh, then they will provide a basic license. And state license given when the particular turnover or particular expense of a particular lab is 12 lakh to 20 crores. And central license is provided when there is more than 20 crores. So you need to also remember what is basic license, what is state license, and what is central license. And you need to also remember WHO, FAO, Codex, ISO, who is their current chairman, where is the headquarters, you need to remember. And ISO, NABL, NABL is a lab in India. NABL, you need to remember the NABL full form, like National Accreditation Board for Testing and Collaboration of Chemicals. This provide ISO certificate. You need to remember ISO 17025 certificate and ISO 15189. Never confused with that. ISO 15189 is a certificate they provide for medical lab, and ISO 17025 certificate given for pesticide testing and calibration. So you only remember 17025 ISO, that is for pesticide testing. And what is bioconcentration? One thing is important. When the concentration increases in body, it's called bioconcentration. And when it comes in food chain, it's called biomagnification. And if the metabolism and excretic of pesticide is low, then accumulation is there, bioaccumulation occurs. So one thing you need in this particular unit, mainly important for particular agencies, labs, what are the certificates they are providing. This is much more than enough in this particular unit. Coming to natural resource management, this particular unit we study in our soil science major group. So we need not to study from chemical points of view, because all these are major chapters in our soil science, and we study in a very detail of this particular. So you do not concern about that something chemicals in chemicals it is given, so something else will be there. Same thing is there, same conditioner, same nitrogen use efficiency, same thing is there. Nothing new in this particular unit. Next is environment, pollution, implication, and remediation. This is unit, and in this 2 points is very important. how the pesticides are impacting the soil functions, soil quality, soil health, and what is persistent organic pollutant. You need to remember dirty dozens. Dirty dozens are also called, these are also called persistent organic pollutants. They are 12 in number. So you need to study, you need to remember what are those chemicals and how they are affecting the different soil properties, how they are affecting different soil functions, how they are affecting the microbial properties, how the transformation of particular elements is affected by these chemicals, how root growth and shoot growth is affected. So you need to study in this particular group. And also the chemicals we are applying, is there any chemicals and after their metabolism they are releasing in atmosphere, like methyl bromide, as I have already told, they have bromine. They generally, when they metabolize, bromine is released and they deplete the ozone layer. So you need to remember in this particular unit, ozone depletion. So this is all the units I have discussed in very brief, that what you need to study in those particular units. And these are the books you need to refer, like Principle of Pesticide Chemistry by S.K. Handa. You will get enough of matter in this particular book. Textbook of Organic Chemistry by Bhal and Bal. Organic Chemistry NCERT, and you study just 2 chapters in there, from there, like biomolecules and synthetic polymers, and one is objective from IARI only. 2 seniors have written that, like Objective Agriculture Chemical by Arkadya Mukhopadhyay. This is a collection of good questions is there collection. But before that, you study Principle of Pesticide Chemistry, Textbook of Organic Chemistry. You need not to study the whole book. First, go through the syllabus and select the topics, and do not dig much more deeper in any particular topic. You need to cover horizontal. You find your topics, and if you're not getting the topics, you just take help of internet, because you will not get all the topics of this particular syllabus in any book or any 4 or 5 books. You need to take help for that particular internet sources. But this will help. You will get it in 12 to 13 question. You will get 9 to 10 easily after reading all these point of view. So this is all for chemical point of view. So if anyone asks something, you can interact. Hello.
Sir. Yes, sir.
If you people want to ask something related to chemicals, anything, you can ask. Look, chemical is little bit difficult for soil sciences student, because you have to remember a lot of names in this particular section. The mode of actions and different chemical name is very, very difficult little bit, but after reading 4 or 5 times, you will get it. And make a short notes while preparing for any exams in this particular, because they will never ask in very much deep. So you need to cover horizontally. But if you have any confusion, anything you have to ask, you can ask.
Chat box.
Can't hear. Can you please? Hello. Hello, Suman.
Sir, please switch on another topic, sir.
Okay. I will continue now. Soil quality, soil health, and
Yes.
soil resistance, and soil quality indicators.
Yes,
Okay.
sir. If any question is coming in chat box, we reply after.
Okay. Text me. I will send. Okay. So the next point is, and this is very small topic: soil quality, soil health, soil resilience, soil resistance, and soil quality indicators. So basically, what is soil quality and soil health? The soil quality and soil health, these 2 terms are mostly used interchangeably. the two terms like soil quality, but scientists refers soil as a soil quality and producers refer to it as a soil health. So you need to remember what is the definition. If it not very much question asked from this particular topic, but you need to remember what is definitions. Like it is the capacity of a specific kind of soil to function within ecosystem and land use boundaries, to sustain biological productivity, maintain environmental quality, and sustain plant, animal, and human health. Soil quality has two aspects, like intrinsic part, inherent capacity, and dynamic part, influenced by user. Intrinsic property, the intrinsic capacity of any soil, it's depend upon mineralogical composition. What is the mineralogical composition of particular soil? What are topographical features? And what is the particle size distribution? That is almost static and usually so a little change over time. And in case of dynamic part, which is based on the management of particular that depend on particularly management. So that's why it is called dynamic. Like soil organic carbon, these all are very dynamic. So soil quality has two aspects, inherent and dynamic. Now coming to soil health is defined as it's a static state of dynamic equilibrium between flora and fauna and their surrounding soil environment, in which all the metabolic activities of the proceed optimally without any hindrance, stress, or impedance. Soil health and soil quality. Sometimes we generally confuse these both terms are very confused. Like soil quality is same and they are synonymous, but soil quality is usefulness of soil over a long time of scale. But when we consider as a what is soil health, it is seen as a particular point, as a particular state. It's for a shorter period of time. But quality refers to usefulness over a long period of time. So you need to remember the time scale here. This is much important. Now, what is soil functions? Generally, we see soil do not function like that. One function now, another function later, but the all function in soil is going through simultaneously. There is a production of biomass production, then filtering, buffering, and transformation. Like filtering of buffering capacity, they maintaining pH in soil, transformation action. Source of biodiversity is there because enormous diversity is present of different bacteria, fungi, all those. Infrastructure development, roads, railway, and these are source of geogenics, and many of the minerals from soils we get, and cultural heritage of landform and paleontological or archaeological traces is there. These all things, these functions can be optimized, and soil if perform very well, so the plant and animal productivity will be higher. They will maintain and enhance water and air quality, support human health and habitation. The question is never asked from this part, what is soil function? How how its impact on different things. But what question asked according to Carlin, they have asked many a times, what are the 5 major soil functions according to Carlin? You need to remember this, Carlin 1997, and you need to remember all these functions: sustaining biological diversity and productivity, regulating and partitioning water and solute flow. Filtering is important, filtering of different pesticides and all these things, water, because there is a absorption process. So soil, via the cation exchange capacity, they absorb different chemicals in soils, they filter out, and the other things reaches in the soil profile. Buffering, the resistance to change in pH or in different things, and degrading of different chemicals in soil by microbial action, immobilizing different, like cadmium is very toxic, and different toxic metals. So they, by formation of chelate or by different things, they immobilize via soil organic matter. They also detoxify organic and inorganic material. We have already discussed in that particular unit in chemicals. This is one of the major function of soil, and some of the, like including industrial and municipal byproduct. These all things you need to remember. Storing and cycling nutrients and other elements within the earth biosphere. We are very well aware that the soil store nutrients, they cycle nutrients, and mainly nitrogen, phosphorus, potassium, all these things. And they provide support for socioeconomic structures and protection for archaeological treasures associated with human habitation. So you need to remember all these 5 points, because sometimes the question has been framed like which of the following is not a functions of Carlin according to Carlin, or what are the major functions according to Carlin? 1, 2, 3, 2, 3, 2, 4 essay. You will get like that. So you need to remember this. The earlier slide is not much important, but this slide is very much important. Now, what is soil resistance and soil resilience? Now, why there is a need of particular soil resilience? Soil resilience term has been introduced to create a common theory that describes the reaction of a particular soil over a range, or when there is a disturbance and when there is an impact of disturbance. So anything which is performing any change from normal pattern or functioning of an ecosystem is there. So we need to study. So the terms soil resistance and resilience has been introduced. So soil resistance, the definitions are very important in these 2 cases, soil resistance and soil resilience. Soil resistance is the capacity of soil to continue to function without change throughout the disturbance. I mean, there is an application of particular pressure. You are adding more and more fertilizer. There is acidification of soil. So how long it can resist, long it can resist the pH, how long it can perform all these activities is called soil resistance. And the magnitude of decline in particular soil resistance depend upon its intrinsic properties, and this defines the resistance to change. If a soil is showing a very little change, it is showing high degree of resistance. And if it is changing frequently after application of fertilizer or any other amendment, so that particular soil so low resistance. And soil resilience mean this is after disturbance. This means ability of a system to return after disturbance to a new dynamic equilibrium. I mean, a system is disturbed. If a system is disturbed, so in how longer time it can regain its activity and it can perform in a similar way. So resilience is that capacity to bring back its original function. So resistance and resilience, these are the 2 key measures of sustainability, and both are dependent on soil types, vegetation, climate, land use, and disturbance regime. This is very much important here. And now coming to soil resistance, these three formulas are very much important when coming to theory point of view: soil quality, soil resilience, and soil resistance. So basically, soil resistance is C by A. C is the level of soil function immediately after disturbance, and A is the pre-disturbance functioning capacity of soil. It has been asked multiple times in various exams. So you need to remember what is soil resistance and soil resilience. Soil resilience is function of soil quality, land use, and management. This is very much important. It has been also asked many times in many exams. Soil resilience is function of basically this change, this land use, or this option will be changed, or this option will be changed. So you need to remember all these things. And what is soil quality? Soil quality, which is function of water capacity. Water capacity, I mean, there should be non-limiting water range. You need to remember this non. This is very much important. A structural index, rooting depth, charge density, or measurement of cation exchange capacity, because cation exchange capacity of soil governs the nutrient transformation. Major nutrient transformation is in soil. Nutrient capacity, nutrient supplying capacity of soil. If the number of clay content is higher, so basically the nutrient supplying capacity of soil is also higher because of the cation exchange capacity of soil will be higher. Biodiversity, because soil biodiversity is one thing that manipulates other function of soil, like they manipulate the physical and chemical function of soil. So this is very much important. Soil resistance and soil resilience both are dependent on soil quality. So you need to remember this. And you need to remember all these three formulas. Now, what are the soil quality indicators and soil functions? So some of here I have mentioned some of the soil functions, and here are some of the indicators. So basically soil function, biodiversity function, like indicators like organic carbon, nitrogen, pH, aluminum, bases, EC, crust, root restricting layer, erosion, sedimentation, plant available water need, weed species and density. These are the indications of biodiversity production. Because if organic carbon is basically a substrate. It is a substrate for microorganisms. So if the organic carbon is low in any particular soil, so automatically the biodiversity will be reduced in particular soil. So organic carbon must be high. Nitrogen, similarly, there is a particular optimum ratio in a particular soil. As the organic carbon increases, likewise nitrogen percentage also increases in soil. But both are in a stable equilibrium or in a dynamic equilibrium in a particular soil. pH affect the richness of particular biodiversity. Like acidic pH is required for fungi, is near neutral to slightly alkaline for bacteria, and above dry soil and for actinomycetes. So this is pH also very much important. Water and solute flow. For water and solute flow, tillage is very much important, because tillage manipulates the physical properties of soil. To flow water, there must be particular conductive, so water will flow. Aggregate stability, earthworms because they they make water channels in soil, they increase the porosity of soil, bulk density. These are very related to this particular things. Filtering and buffering. In the soil function, filtering and buffering, the indicators is basal respiration, organic carbon, texture, microbial biomass, cation exchange capacity, chemical loading, herbicide residue. These all are related to filtering and buffering. Now coming to nutrient cycling. In nutrient cycling, there is organic carbon, nitrogen. Again, cation exchange capacity is very much important here, because without cation exchange capacity, none of the reactions is possible in soil. That's why it is called that after photosynthesis, cation exchange capacity is very much important reaction in soil. And one minute, something is repeated over here, I think. No, it's okay. Now, particulate organic matter. This is particulate organic matter, potentially mineralizable nitrogen. Potentially mineralizable nitrogen, I mean the nitrogen which can be mineralized by particular microbes. There's a potential in soil. When you in a lab experiment, it's already studied that in Subia Asija method, the nitrogen that is mineralizable, we already see it. But when you incubate the soil, there is a conversion of some more nitrogen, another pool of nitrogen to potential mineralizable nitrogen is there. So this is also important. And for structural support, there is a soil structure, texture, bulk density, landscape position, and aggregate stability. This is very much important in case of structural support. So these are soil quality indicators, parameters. Now, factor affecting the soil quality. Why soil quality? We are more concerned. What are the factor effects here? So there is a wide gap between nutrient demand and supply, because what plant need, and we generally do not go for soil test. When we do not go for soil test, you will apply either blanket application, or you will apply based on earlier recommendations. So if soil testing is not there, and you are not knowing how much hunger is soil, so you'll apply that. And the choice of crop also affect. Like some crops are very much exhaustive, and we apply nutrient in particular dose. And if you talk about potassium, like many times it has been seen, potassium is a nutrient which required in maintenance dose. Potassium, there is a pool of potassium, like it's come from non-exchangeable pool, then it's come to exchangeable pool, then it's come to soluble. Here non-exchangeable, before non-exchangeable, it comes from mineral composition. Here the mineral to non-exchangeable pool, the reaction is purely irreversible, and here there is a irreversible reaction. So if you not apply potassium in particular field, so what happens, the mineral potassium come into non-exchangeable. The mineral potassium come to non-exchangeable pool. The mineral potassium come to a non-exchangeable pool, and the reaction is irreversible. So basically, the structural potassium is losing from minerals. Structural potassium is losing from minerals. So the stability of particular soil is getting decreasing, and this is irreversible process because a particular mineral formation takes a lot of time, and within a human lifespan of it is not possible. So we need to maintain a particular uh, uh, particular maintenance this application in case of potassium. And other nutrients like nitrogen and phosphorus, this we have to apply as per the demand of the crop. But potassium is seen that in few decades that it has gone quite deficient, and although the Indian soils are rich in many places, rich in potassium, but they are showing deficiency because of that. High nutrient turnover in soil plant system coupled with low and imbalanced fertilizer use. This is one more point. Like the soil, the plant is requiring like 220 kg, and you are applying 120 kg nitrogen. So the remaining nitrogen will be either taken from soil or from different pool of nitrogen, or maybe the potential pool will be utilized. So there will be create an imbalance, and any imbalance will not only hinder nitrogen only, because this also affect the uptake of phosphorus. This will also affect the uptake of potassium. So you need to understand this. There is emerging deficiency of secondary and micronutrient due to improper use of inputs such as water, fertilizer, and pesticides. We already know the micronutrient deficiency is only 2 to 10%. Only 2 to 10% is there, and in case of sulfur only there is a 10% deficiency. Others in case of 2% only is there. So most of the times what happens, the farmers do not apply micronutrients; they apply only major nutrients. So there is a deficiency of these micronutrients, so they affect soil. Because the plants what it takes from soil pool. Insufficient of organic inputs. Many times in going for higher yield, what we do, we apply mainly inorganic inputs, but there must be a judicious use of organic inputs, so that the plant can perform well and the soil quality can be made and sustainable production can be taken. Acidification and aluminum toxicity. Acidification, what cause? Acidification is the root cause of any chemical transformation, and the pH alone all governs the nutrient cycle. Aluminum toxicity, it directly affects root growth. It directly affects the root growth and reduces the root growth, and it decreases the productivity of roots. Now, development of salinity and alkalinity in soil. Salinity and alkalinity is a vast topic, and it's how it's impact the plants. And here it has been seen that the salinity alkalinity is increasing. The area is already increasing from this much hectare to this much hectare. So this is also a problem. Development of adverse soil conditions such as heavy metal toxicity. Heavy metal toxicity is not only concern of soil, but it's when it's coming to grains, it's accumulating in grains like cadmium, arsenic. These are heavy metals which are coming to grains and causing illness to humans. So heavy metal toxicity is a major concern. So breeders are generally doing these things. They are modifying the plants as per so that they can accumulate less, but this is not just only an option. There must be a judicial use, because when you add a phosphoric fertilizer, it might contain little bit of cadmium or arsenic residue is there. So we need to understand this. And disproportionate growth of microbial population responsible for soil sickness. I mean, when soil is sick, I mean the particular there is two types of microbial population. One is healthy and one is unhealthy population. So there must be a particular proportion in root and as well as rhizosphere. There must be a particular proportion. So when the proportion is get disturbed, so soil is unable to perform a particular function in that way. So that affects soil quality. And natural and man-made qualities, like natural and man-made calamities, like natural, we cannot check natural, but the man-made activities we can stop. And the man-made are also the leading activity that are mainly responsible for disturbance of that soil. So these are the factors that affect soil quality. This is not, and so this is much from in this particular topic, because this particular topic was very short. So anything you can ask. This is all for today.
If any question to any participants, please ask to the sir.
Or you can also text Suman. You can collect questions from a participant if they are not
Yes,
ready
yes,
to
sir.
ask. I will provide the answers already. Yeah?
Yes.
And if you need the detail of that chemical in written form, I will also give that, which particular topic is important and what is asked. I will give that in written form. Okay.
Okay.
So should I unshare the screen?
Yes, yes, sir. You answer the screen, sir. Sir, there is one question also in the chat box, sir.
Let me see.
Mamta ma'am, just to thank to you, sir. There are no questions, sir. Only one Thanksgiving information.
Okay.
So thank you, sir, behalf of all the students and the MPKB Rauri, and behalf of our HOD, sir, because he is busy in the one meeting, so they not join. And today also some another meeting program, another NBSS and UP organized one program in our university. So some students engage in this program. So there is some less students join this program. So thank you, sir. Thank you very much, sir, for giving this precious time to us. Thank you.
Okay. Thank you so much. So I can leave?
Yes, yes, sir.
Okay. Thank you.
Student, there is a one class for 9 o'clock from Thakuria, sir. Please join at the time. There is a one class of Thakuria, sir, of 9 o'clock today morning. Please join at the time.
Morning, yeah, evening.
No, morning. Tomorrow, not today. It's tomorrow. Tomorrow morning, there is a one class of Thakuria, sir. They taken the rest of the his portion on the nutrient cycle.
Okay, sir.